too much triglycerides that you would find in plasma. I mean, same. Match together. Thankfully, it is my only class, so like all I gotta do is look for this. And one other question will start till the week of the 25th. Oh, I wish I graduated. I'm graduating, that's why I have 6 credits left. I wish I would. No, you don't, because I was supposed to graduate in December, but my advisor apparently discounted my credits. So he got my hopes up for a whole year about me graduating in December, but nothing. Well, like, my husband's so far, knock on fake granite, it turned out. I think it's finally less. My other question is, this is next to my hair. Yeah, I think same.
Sometime in the weekend.
Damn good weed. Somebody needs to snatch it so we can verify it.
In all honesty, if I do think I'm going to score better, does not mean I'm going to blatantly
Like even with this is my only class, it's still pretty stressful.
for life.
Because it's like so much information.
4 years, if you
And then bringing back 4 hours.
taking on a baby.
I'm trying to remember what
No, that doesn't. It's facilitated diffusion, so it doesn't require amputation.
some of the graphical stuff I'm looking at. And I'm just like, man, we'll do it now.
To be consistent, what can I say? Just to be thorough. I'm gonna watch, detail.
Josh, Jesus texted you about that Taysachs question.
Yeah.
Is that on the exam or no?
No, it won't be on the exam. Yeah. Alright, so it's still recording.
I mean, like, so far it hasn't been like so good.
Oh, no.
Like, you're still able to read it, I read like don't remember, yeah. Like, I didn't remember what it was. Exactly. Like, if you put it in front of me with a bunch of options, I can tell you which one it is. I can't tell you off the top of my head.
Alright, so a lot of, if you were at the exam review, a lot of what we're going over today will sound a lot similar to what I went over last night. I may give a little bit more detail about certain things, but a lot of it's going to sound similar. So, Chapter 8A and 8B. 8A has to do with lipids, the different categories of lipids, and 8B has to do with the cell membrane and cell membrane pumps and things like that. So, going down 8A, you'll be relieved to know that you really don't need to know the very specific names of all these different lipids. So you don't need to know lauric acid, myristic acid, whatever. You don't need to know the specific names of these things. It is way more important for you guys to be able to distinguish these lipids from each other, distinguish the categories of lipids from each other. So for example, and we'll get to it, if you're given like this structure, you should at least be able to tell me what category of lipid it is versus maybe this structure. And if you're at the exam review, you guys should actually very easily know what type of structure this one is. It is a ganglioside, exactly. And we'll get to all of that. So each one of these has certain features that make it a little bit easier to identify what type of lipid it is. And that's what we'll be mostly focusing on today. I give you a couple of extra questions and things on here to help reinforce some of that. But again, the most important things will be you have to be able to identify the general features here. So, we'll start with fatty acids. Fatty acids are really easy to identify. Let me look for a nice picture. Is there a zoomed in picture somewhere? Yeah, here we go. Fatty acids are very basic and easy to identify. There aren't any complicated functional groups on them. You have this carboxylic acid side, that's why they call it a fatty acid, and you have a long hydrocarbon tail attached to it. So as long as you have those two features and nothing else complicated, you know you're dealing with a fatty acid. Now, there are two categories of fatty acids, of course, that you guys probably know from biology or earlier courses: saturated fatty acids and unsaturated fatty acids. Saturated fatty acids are easy to identify because there's only single bonds in them, while unsaturated fatty acids have at least one double bond in them. Now, this doesn't really illustrate it, but in nature, what type of double bond will you see in an unsaturated fatty acid? A cis double bond. You're not going to find any trans double bonds in naturally occurring fatty acids, so that's very important. Don't worry about memorizing melting point and what fatty acid has what melting point. It is more important to understand what features of a fatty acid influence the melting point of that fatty acid. So, in this example here, we'll start with this. You see the number of carbons here listed on the left and the melting point for each of these fatty acids here. What is the link? One question could be, what is the link between number of carbons and melting point? Well, we can see a nice trend here, and that the more carbons you have, the higher the melting point you have, which is which makes a lot of sense, right? If you have more carbons, you have a greater attractive force between these carbon atoms. In fact, what intermolecular force is going to keep these hydrocarbon tails together? The hydrocarbon tails will be held together via London dispersion or van der Waals forces, because all you have are carbons and hydrogens in these tails. So when you add more carbons and hydrogens, that's the only force you are strengthening. Yes, at the very head portion of this, you will have hydrogen bonds, but the only difference between these fatty acids when you compare them to each other isn't the head, but just how many carbons are in the tail. So increasing the number of carbons increases the attractive force between the tails of these fatty acids, thus increases the melting point. You need more energy to break it. So that's one factor that affects the melting point of a fatty acid. The other factor is the presence of at least one or more double bonds. So here are your unsaturated fatty acids, and you may immediately notice that all of these here have a really low melting point relative to that first list we were looking at. That first list of fatty acids we were looking at had relatively high melting points because they were all saturated fatty acids. They were all single bonded fatty acids. Your unsaturated fatty acids, as long as it has at least one double bond, greatly affects the melting point of these unsaturated fatty acids. It's a lot lower. And the reason why is, if we look at a picture of unsaturated fatty acids here on the right, notice it's a lot harder to pack them close together because all these double bond bends here prevent you from packing these adjacent fatty acids close together. Contrast that with saturated fatty acids without any double bonds in them. Notice how close you can pack them together. Therefore, saturated fatty acids have a higher melting point. There used to be, in old semesters, a question on the exam where they say, which is more likely, something on the order of which is more likely to contain saturated fatty acids, a cold weather plant or a tropical plant? Well, saturated fatty acids have a higher melting point. Therefore, saturated fatty acids you're most likely going to find in a tropical plant because it is a warmer environment. Unsaturated fatty acids you're therefore more likely going to find in a cold weather plant. I believe some examples he gives of each of these is like coconut plant for saturated fatty acids and like olive oil or something for unsaturated fatty acids, but you don't need to know that anymore. Just the most important thing is the two factors that affect melting point: number of carbons and double bonds. Okay, so yes.
He did say in class that on the exam he will ask something about like Spain to choose the one that has more double bonds.
To choose the one that has more double bonds. I haven't seen anything like that. I'd have to get the full context of that, but that doesn't sound familiar. No.
It was because it was tropical or
The tropical versus cold weather thing? Yeah, so if that shows up, just understand that all he's asking you is what type of fatty acid are you most likely going to find in certain environments. Well, we just went over saturated fatty acids have a higher melting point, therefore the reasoning is you're going to find them in tropical plants where it's warmer. Unsaturated fatty acids you're going to find in cold weather plants. Yes?
So the more double bonds that an unsaturated fatty acid has, the lower the melting point?
I won't actually go as far to say that. It's just the presence of double bonds will lower the melting point. I don't think it is 100% true that more double bonds means less melting point. It is simply the presence of double bonds will lower the melting point compared to if there's no double bonds. All right, so moving along. Oh yeah, you may be given any pictures of these fatty acids. Like you may be given pictures like this or like this one up here. You won't be asked to name it, but there are a couple things they could ask. For example, a vocab or type of memorization question is which fatty acids are your essential fatty acids? So first, what an essential fatty acid is. An essential fatty acid is one that our bodies cannot make. We don't possess the mechanisms to synthesize these fatty acids ourselves. And so essential fatty acids we have to get in our diet. There are only 3 of them, and they all sound very similar to each other in terms of name: linoleic, alpha-linoleic, and gamma-linoleic. So it's easy once you memorize that, and you know that those are your essential fatty acids. They're all unsaturated fatty acids, but then what's unique here that hasn't been shown anywhere else in the notes yet, or maybe other places actually, is this numbering system. So if you're given a unsaturated fatty acid you've never seen before, will you be able to figure out certain properties based off the numbering system? So, let me break down the numbering system. Whatever unsaturated fatty acid you get, the very first number that you see is going to just tell you the number of carbons that unsaturated fatty acid is composed of. What is the second number going to tell you about that unsaturated fatty acid? Number of double bonds. And this one here is also called the omega number. The definition for this number is a little bit long, but all the omega number tells you, and let me look for a nice example. Where's DHA? That one's a nice one. All the omega number tells you is the number of carbons you travel from the tail end of your fatty acid till you hit the first double bond. So let's look at this example up here. The omega number of DHA is omega number 3. And why is that? Well, if we start at the very end, 1, 2, it takes 3 carbons before we hit the double bond at the end there. So it has an omega number of 3. So you can guess that omega number 6 probably means that for this particular unsaturated fatty acid, you have to travel 6 carbons from the end before you hit your first double bond. So 3 numbers here are very important. If you're given an unsaturated fatty acid you've never seen before, you'll still be able to answer questions on it based off dissecting the numbers that are given to you. Okay. All right. This mechanism, well, it's kind of the start of the mechanism of, there's a mechanism for inflammation. And I went over at the exam review, and I will, at this point, I'm not going to go over it in detail. I'm going to point out the important slides that you should review. But this is one of your possible extra credit questions. You may get a question that asks, describe the inflammatory pathway. Basically, just conceptualize, describe what is going on. You don't have to draw out all these molecules and the names of each one of these things. You don't have to do anything like that. So in fact, let me jump to it. Since, yeah, this slide and Yeah, actually, it's just really, this slide is probably the most important slide when it comes to answering that question about the inflammatory cascade. The caption here on the left describes exactly how it starts and how it ends and how it is inhibited. So the main goal, the main purpose of this pathway is for you to produce what are called cytokine molecules, such as leukotrienes and prostaglandins. These things cause inflammation, redness, swelling, and increased temperature. We have designed drugs to inhibit this pathway. You might recognize the name of some of these drugs: aspirin, ibuprofen, Vioxx, and Celebrex or Celebrex or whatever it's called. And they inhibit various enzymes in this pathway. So if this ends up being your extra credit, you need to describe everything that is written here on the left here, and what the purpose of this pathway is, which is to cause inflammation. You give me all that information and I'll believe that you know what you're talking about. Question?
Do we have to like write out the entire name of like PLA2?
No, just the acronym is fine for each one of these things, that's fine. And you don't, and I'll even be generous, you don't have to tell me specifically that ibuprofen inhibits both enzymes, but Vioxx only inhibits COX-2. Just list the drugs and tell me they inhibit the COX enzymes, and that'll be enough detail for me to give you credit there.
C2 position and all that in the beginning.
No, just tell me that PLA2 releases arachidonic acid from phosphatidylglycerol and you're good. In terms of multiple choice, among this stuff, probably the most important information, if this shows up as multiple choice questions, are the enzymes themselves, the ones that regulate this whole pathway, COX-1 and COX-2. These are the ones that actually catalyze the reactions of the inflammatory pathway. And inhibiting these enzymes, we've designed various drugs to target these things. Yes?
What spelling count for phosphatidylglycerol?
Just make it close enough. And again, I will be pretty lenient because this is a fairly complicated piece of information. Just show me enough that you know what you're talking about. Yes, question.
So the drugs inhibit COX-1 and COX-2, not the weird name?
Yeah, not PLA2, right? Yeah, no. The drugs are, we've only been able to design drugs that inhibit these two enzymes. So they're kind of like in the middle of the pathway rather than at the beginning. And that's still fine. If we inhibit something before the end, it'll still inhibit inflammation. Yes?
What is COX-2?
Yeah, so they're saying the side effects of inhibiting each one of these things. So inhibition of COX-1 ends up producing very bad side effects like kidney problems and stomach problems, and inhibiting COX-2 sometimes leads to heart attacks. So it doesn't mean this is a common thing. These are just known side effects of inhibiting these enzymes. So in terms of differences between COX-1 and COX-2, they're listed right here. COX-1 is the homeostatic response for inflammation, and COX-2 is for emergency response for inflammation. Although technically both are kind of used simultaneously, realistically speaking, but these are the technical definitions for these enzymes here. Okay, so we got to move on. All right, let me go back up here because I think I skipped two categories. All right, the next category of lipids are triglycerides. And I've already had some people ask me about a question referring to triglycerides on the practice exam, so I will point out some very important information about this. Your triglycerides are your energy storage lipids. These are and they're the only category of lipids that are able to store energy in a form that you can later extract energy out of. All those fatty acids we were talking about earlier actually exist in the form of a triglyceride when we're talking about energy. Let me pull up a picture of a triglyceride. Here's one here. A triglyceride is a glycerol molecule bonded to 3 separate fatty acids. They can be the same fatty acid or they can be completely different fatty acids. It doesn't matter. So fatty acids don't float around by themselves normally in the cell. They're usually attached to a glycerol molecule at the very least. So triglyceride is an energy storage molecule. Will you find triglycerides in cell membranes of various different cells and tissues? The answer is no, because it does not serve any structural purpose. A triglyceride is an energy storage molecule. Triglycerides you're very often going to find in fat cells, and There's a scientific medical term for that. It starts with an A. Anyone know?
Adipose.
Adipocytes or adipose or something like that. Those cell types, more than any other cell type in the body, are gonna be abundant in triglycerides, just in that cell. As for other stuff, they give you more information on that, but most of that is too specific for the exam, so I'm just gonna skip over it. The next category of lipids, and again, it's more important to know category of lipids rather than specific detailed names. The next category is glycerophospholipids. Let me just jump to a picture so we can take a look at it. So each one of these things listed here are glycerophospholipids. They're very easy to identify because they look a little bit like triglycerides, but they're slightly different. You see a glycerol molecule in the middle here, that's 3 carbons. You see 2 fatty acid tails attached to it. And what's special about a glycerophospholipid, and this is the first lipid category you'll encounter, that has a phosphate head. What I mean by phosphate head is there's a phosphate here and some special functional group attached to that phosphate head. There's several examples of glycerophospholipids. This one is phosphatidylcholine, and that's called a choline group here. This is phosphatidylethanolamine, ethanol with an amine group attached to it. Phosphatidylserine, there's a serine group. Phosphatidylglycerol, that's an extra glycerol here. You don't have to know the specific glycerophospholipid, but if you're given any one of these, you need to be able to tell me that it belongs to the glycerophospholipid group. And notice they stopped drawing the tails here. These are still fatty acids, and they can be any 2 fatty acids. It doesn't have to specifically be these 2 fatty acids. It could be any 2. Yes?
So if we get a picture of one of these, and all we have to know is glycerophospholipid.
Yep, is that they're glycerophospholipid. You have to know basically that they're not triglycerides, that they're not sphingolipids, just what category does it belong to. Yes?
Would we be getting names like phosphatidyllethanine or like...
You'd be given a picture rather than a name. You'd be given a picture.
So it gives us names of like a bunch of them that say which of these like does not belong in like,
No,
whatever.
not for this exam. No, you would just plainly. There's quite a lot of like picture type questions on this exam because you have chapter 7, which has monosaccharides and disaccharides and polysaccharides in it, and you have chapter 8A, which has all those different lipids types. So there will be quite a few where they give you a picture and you're supposed to answer what are you looking at, which in some ways is actually pretty easy.
Are they all saturated?
These here, they don't have to be, no. It's just in this example, they all happen to be saturated. Question?
There's almost 2 fatty acids.
2, yes. There's only 2 fatty acids attached to these, not 3 like in triglycerides. There's only 2 of them. And you can see why, is because in this glycerol molecule, the 3rd carbon here, instead of having a fatty acid attached to it, it has this phosphate head attached to it instead. Okay. All of these you can find in a cell membrane. So glycerophospholipids are components of cell membranes in all types of tissues. Okay. All right. We already talked about that stuff. All right. The next category is very similar to glycerophospholipids, and it's because there's just a slight alteration in it. Are ether glycerophospholipids. And even amongst this, the only one you have to know about is specifically plasmalogen. It's the only example of an ether glycerophospholipid they give you. So this one you should recognize by name specifically, and that is plasmalogens. So let me go to a picture of well, this is an ether glycerophospholipid. Ether glycerophospholipid, again, small little differences, but once you know the differences, they're actually pretty easy to recognize. Here's a glycerol. Here are 2 fatty acids. They got lazy and they didn't draw it all in, but there are 2 fatty acids here. One of them is still an ester-linked fatty acid, but the other one is changed. The other one is now an ether link. So let me contrast that back with glycerophospholipids so you guys can conceptualize this. Look at the 2 tails here. They are both esters up here, ester linkages. And now for ether glycerophospholipids, one of the tails is now an ether linkage. That's it. Everything else is the same. Polar phosphate head, glycerol group, whatever. Those are your ether glycerophospholipids. Sometimes he has a picture of plasmalogen. There we go. Plasmalogen, you see, fits the same property here. You have a glycerol, you have 2 fatty acid tails, one of them is ester linked and the other one is ether linked. This one just happens to have an unsaturated fatty acid in it. It doesn't matter, it is still an ether glycerol phospholipid. And of course you have your polar phosphate head. Note that plasmalogens you're going to find are very abundant in the cell membranes of heart tissue and heart cells. And again, I bring that up because plasmalogen is the only example of ether glycerol phospholipid you have to know. Yes, question? Yes, that is what an ether glycerol phospholipid as well. Is by definition, when you look at the 2 fatty acid tails, one of them is ester linkage and the other one is ether linkage. Again, to help you conceptualize that, I suggest contrasting that with regular glycerol phospholipids, where you will see that both of these are esters. So an ether glycerol phospholipid is one of them is replaced with an ether group instead of an ester group. Okay. So let me move along. The next category of lipids are sphingolipids. And conceptually, sphingolipids are all built off of a backbone called a sphingosine backbone. But even more specifically than that, they're actually built off the second molecule listed in this picture here called a ceramide. Sphingosine is what they say it's built off of, but practically speaking, like visually speaking, you're going to recognize ceramide a little bit more often than you're going to see sphingosine. So it is my suggestion that when you're studying sphingolipids and you're trying to figure out what structure, what given structure is a sphingolipid, look at ceramides to start with. So ceramides are what all your sphingolipids are going to be built off of. So what features do we have here? Well, we have a glycerol-looking molecule here, and we have a tail here. It's not a fatty acid though, which is interesting, but we have a tail here, and this is unique to sphingolipids. Is this amide group here? You're not going to find any other type of lipid that has an amide group attached to this glycerol here. Okay. Now let's see what the actual sphingolipids are, because ceramide is just a base for sphingolipids. First example here is something called sphingomyelin. We see right here a ceramide group, and how do I know it's a ceramide? Well, we have this tail, we have the glycerol, and we have the special amide group here that nothing else we've learned has. So that's the ceramide base here. So what a sphingomyelin is, which is one of your 3 sphingolipid categories, what a sphingomyelin is one with a polar phosphate head attached to a ceramide. That's it. So once you learn the ceramide structure, are able to identify the ceramide structure, the way the head changes tells you what type of sphingolipid you have. If there's a polar phosphate head attached to the ceramide, it is sphingomyelin. You may recognize that term in probably some other classes like an anatomy class or human morphology class. Sphingomyelin is a very important component found in neural cell membranes, right? Have you heard of the phrase myelin sheaths? Those are those things that can help insulate and conduct electrical signals in a neural cell. And the reason why they're able to do that is because a myelin sheath is composed of a bunch of sphingomyelins. So that's to help you kind of memorize what sphingomyelins are and where they come from. Okay, there's 2 other sphingolipid types. One is called a cerebroside. So what a cerebroside is, let's take a look. Here's my ceramide again. And all a cerebroside is instead of a polar phosphate head, we just have a singular monosaccharide attached to it, just one sugar attached to it. And that's it for cerebroside. After that, we have ganglioside, where all the way down here, in this crowded picture here, we have a ceramide still, but instead of one sugar, we have multiple sugars attached to it. That tells me that this is a ganglioside. A gang of sugars is how I memorize it. So your 3 sphingolipid types are sphingomyelin, which we see is made of a ceramide plus a phosphate head, cerebroside, which is made of a ceramide and a single sugar, and ganglioside, which is made of a ceramide and a bunch of sugars. And then you're done with sphingolipids. He did mention Taysachs disease, and in previous semesters he would ask what Taysachs disease is or what causes Taysachs disease. I'll give it to you here, but you won't need it for the exam, is that Taysachs disease is caused by an accumulation of sphingolipids. I forget which one, maybe is it cerebrosides? No, it's gangliosides, whatever. But it turns out there's a lot of diseases that occur because sphingolipids are improperly disposed of, and they build up in the body. They start clogging up your neural cells and lead to a bunch of neural defects. So you don't need to know that for the exam, but that's the context for Taysachs disease. Waxes, you don't need to know about for the exam. Terpenes, you don't need to know for the exam. Cholesterol, you don't need to know how it's synthesized, but if you're given a picture of cholesterol or something that looks similar, as long as you can identify it as cholesterol as opposed to the other categories of lipids we've gone over, that's all you need. And cholesterol should be easy to identify. It's the only category of lipids that has these 4 rings. And now that I brought that up, I will generalize it even more and tell you that cholesterol is a precursor to a bunch of to a lipid category called steroid hormones. So if you are given either cholesterol or anything here, you'll notice very quickly they don't look anything like the other lipids we've gone over. These are, I think they call it a tetracyclic structure or something like that, a 4-ring structure. That is a characteristic of steroid hormones and of cholesterol. You may recognize some of these as being your sex hormones. So be very careful there, is that sex hormones are specific steroid hormones. Not all steroid hormones are sex hormones. For example, cortisol is a steroid hormone that has nothing to do with the other sex hormones. So these types of pictures are indicative of steroid hormones. Don't need to worry about this. These are other terpene-like structures, which you don't need to know for this exam. And that's the end of 8A. Let's answer a few questions real quick, and I'll go on to cell membrane stuff. Let me find some... Yeah, let's go to the most important thing, is identifying these things. All right, let me do it a little bit out of order. Well, what's that? It's a steroid hormone or cholesterol. Either one would be fine. What about this one here? It is sphingomyelin. That is correct. It is sphingomyelin. Let's take a look. We see a glycerol-like structure here, but instead of 2 fatty acid tails being here, we see this one chain here, and the very not easily identified, but the very characteristic structure is this amide group here, and we have a phosphate head up here, so that becomes sphingomyelin. This is easy. What is it? Ganglioside. How about this one? Cerebroside. Exactly. This one is a ceramide attached to 1 sugar. How about this one? Plasmalogen would be fine too, but a more general answer is that this is an ether glycerophospholipid.
It's not unsaturated.
It doesn't have the unsaturated part, yeah, but for the exam you don't actually have to distinguish it that specifically. So this is the more general answer that is correct here, is that this is an ether glycerophospholipid. A plasmalogen just has one additional modification in that there's supposed to be a double bond here, but ether glycerophospholipid is the correct answer. What about this one? Just regular glycerophospholipid, exactly. You notice that these are both ester linkages right here. What about this here?
Ceramide?
That is ceramide, just ceramide by itself. It isn't sphingomyelin or ganglioside or cerebroside. This is just ceramide by itself. And I think those are all the ones that I have here, so that's it. Okay. So again, the most important thing in 8A is being able to identify the different lipid categories from each other. All right, 8B, and I'll try to go through the most important parts here. 8B talks about cell membranes and the components of the cell membrane. Yes, question?
What was the thing you said yesterday during the review about if you have 2 choices and both are correct, pick the more specific one?
Yeah, I can't remember. Was that maybe with sugars or something?
Steroid hormones.
Oh yeah, steroid hormones versus sex hormones. you won't be given a question that specifically asks, is this a sex hormone or a steroid hormone? You would be given like estrogen, and it'll give you like, which of the following categories does this molecule fit in? And the answer would be steroid hormone. You won't have an answer choice that says sex hormone. Oops, someone's got a call. I'm going to hang up there. Okay. Let's see. Continuing along with cell membranes. So, after we talk about lipids, now we talk about the cell membrane part, because this is what the lipids very often come together to form. So, let me find a nice picture of a cell membrane. I think there's an even better one. You know what? I prefer this one. Okay, so cell membrane. We learned way back in like chapter 1 or 2 a term called amphiphilic molecules, right? Or amphipathic molecules. Those are those molecules that have both a polar end and a polar head and a nonpolar tail. It turns out when you combine some of these amphiphilic molecules together, you can form something called a lipid bilayer or a cell membrane, and you see that illustrated here. A cell membrane or lipid bilayer has 3 regions: an extracellular region, meaning outside the cell, an intracellular region, meaning inside the cell, and an intermembrane space, which is very important, and it's the widest portion of a lipid bilayer, an intermembrane space. The extracellular and intracellular portions of a cell membrane are polar, because that's where all the water is located, the water inside the cell and the water outside the cell. So the polar heads have to be able to interact with these aqueous environments. Inside the cell membrane, in the intermembrane space, these are all where all of your nonpolar tails are going to be. So this is a very nonpolar environment. So, for example, if I'm looking at this integral protein here, it is more specifically a membrane protein, but you can classify it as an integral protein. If I'm looking at this here, and I wanted to analyze the amino acid structure of this protein, what category of amino acids do you think is going to be located right here in this integral membrane protein? Non-polar amino acids will be found here, because that's the intermembrane part of it. That is where the non-polar region of the cell membrane is. Therefore, an integral protein needs to be composed of non-polar amino acids here. What about on the extracellular and intracellular parts? What categories of amino acids do you expect there to be? Polar, and even more than that, acidics and basics, because those are charged amino acids as well. So that's very important to conceptualize. So I already mentioned integral proteins. Integral membrane proteins are the ones embedded into the cell membrane. The other category of membrane protein are peripheral membrane proteins, which are illustrated like this. They're very loosely associated with the cell membrane. In fact, peripheral membrane proteins are only able to bind to the cell membrane due to a series of molecules called lipid anchors. So lipid anchors, which are given in way more detail than you need in the notes, lipid anchors are those molecules that bind to peripheral membrane proteins and make sure they at least temporarily stay bound to the cell membrane. There isn't really a picture of a lipid anchor.
Do we have to know like the specific
The types?
Yeah,
No, you used to have to, but not for this exam. There isn't enough time, so any questions like that I've kind of removed. Lipid anchors, you don't need to know the different types of lipid anchors, thioester, thioether, amide, whatever. Don't worry about the different types of lipid anchors. It's fine to know, and you may not be asked about anything like this, but it may be on the final exam, or what is the general function of lipid anchors. I gave you one function already, is that they help link peripheral membrane proteins to a cell membrane. There's a couple other functions as well listed right here. Okay, now we go into the different ways molecules are able to cross a cell membrane. There are 2 categories: passive transport and active transport. So I'll go over the 2 categories first, then we go into specifics. The definition of passive transport is simply that molecules travel with the concentration gradient, meaning physics likes it when high concentration stuff moves to low concentration stuff. So passive transport, whatever form, must have that at least, that the molecules go from high concentration to low concentration. Active transport, by definition, is some type of transport that goes against the concentration gradient, where you take molecules on the low concentration side and you push them to the high concentration side. That is not a favorable mechanism, so active transport mechanisms are always coupled to something to give them energy. The most common thing that will give them energy to perform active transport is ATP, which we've talked about a few times already. We'll get to a couple examples of active transport in a second. So, first type of passive transport is called passive diffusion. Passive diffusion is simply the molecules moving from high concentration side to low concentration side without the help of any membrane proteins. Some molecules can simply just slip in between the cell membrane lipids and just pass out from the outside of the cell membrane to the inside of the cell membrane. In fact, the category of molecules that are able to do this very easily are your steroid hormones. Your steroid hormones, which we learned was a type of lipid, are very nonpolar. Therefore, they can easily diffuse through the cell membrane without the help of any membrane proteins. The other type is, I mentioned it at the review, and I'm not going to be able to go over a practice question of that since we're running out of time. This equation here, you will be using it for one question on the exam. It is on your Chapter 8B PowerPoints. It is called the Nernst equation. You just need to know the modified form in purple here and be able to use it. Basically, if you're given concentrations of an ion, both on the outside of the cell and the inside of the cell, you plug in those concentrations into this formula, and you'll be able to calculate the resting potential of whatever these ions, whatever potential these ions cause. So it's very simple. You're going to be given two concentration values. You plug it in here, and you get your answer.
And where?
It will be given to you. They will be the numbers will be given to you. You just have to know the formula and be able to plug it in. Okay, blah 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 blah. In fact, you may be given any one of these. I can't say which one, but you would be given the numbers, so don't bother memorizing the numbers anyway. Okay. The other type of passive transport is called facilitated diffusion. It's the same thing as passive diffusion, except facilitated diffusion has a bit of an advantage. There's no picture of one. Facilitated diffusion uses cell membrane channel proteins to help molecules pass through the cell membrane. Passive diffusion, there was no help. The molecules had to try to push their way through the cell membrane. Facilitated diffusion still goes with the concentration gradient. It just uses channel proteins to help molecules move through the cell membrane. The advantage of this is that this is a much faster process compared to passive diffusion. Molecules that are able to pass through a cell membrane through facilitated diffusion do so at a much higher rate, which is illustrated here, but don't worry about memorizing these graphs. Okay, now we get into some other vocabulary terms that we can use to describe any type of transport. That is the phrase uniport, symport, and antiport, which are illustrated here. Uniport is simply when a transport protein transports one molecule in one singular direction. Symport is when a transport protein takes two molecules at a time and transports them in the same direction. And antiport is when a cell membrane, when a transport protein takes two molecules at a time but pushes them in opposite directions. Yes?
Are these in active and passive?
Yes, these can be classified to either active transport or facilitated diffusion. These terms only describe the direction that molecules move in relative to each other. That's it. So, the first specific example of a transporter is called the sodium glucose transporter. This is the one example of facilitated diffusion given to you in your notes. So this is facilitated diffusion because what this does is it takes a sodium and a glucose and it transports them down their concentration gradient from the intestinal from the intestine into the intestinal cell. So notice it's transporting two molecules at a time. So what other term can you use to describe this? symport. It's taking two molecules and moving them in the same direction. So sodium glucose transporter is an example of a symport, and it is facilitated diffusion, so it does not require ATP. You may see this phrase, sodium is ATP transported. That is because of the next thing we are going to talk about, called the sodium-potassium pump. This thing by itself, the sodium glucose transporter, has nothing to do with ATP. It does not require ATP. Sodium, though, how did it even get outside the cell in the first place? It was because of the next pump that we are talking about, which does use ATP. Oh yeah, one extra fact here, this drug, Farxiga or whatever, inhibits this pump, this transporter, the sodium glucose transporter, which you can find in kidneys and small intestines. Okay. Active transport. I said earlier that active transport is when molecules move against their concentration gradient. The most important one that you need to know for this exam is the sodium-potassium pump. It does help to know the particular steps in it, which are kind of illustrated here, but in my opinion, looking at this diagram helps a lot instead. So here's the order of events in the sodium-potassium pump, and I go over it also during the exam review. First, the pump is actually closed on the outer surface and open on the intracellular side. Therefore, the only thing that can bind to it is sodium molecules. So 3 sodium molecules bind to your sodium-potassium pump, and they are getting ready to be transported out of the cell. Before that can happen, you need to have ATP phosphorylate this cell membrane protein. So once your sodium ions are bound to this thing, and once ATP phosphorylates it, it is now ready to transport the sodium ions out. What ends up happening is that once it is phosphorylated, the intracellular part of this protein closes and the extracellular part opens up. So all the sodium that was bound to it gets ejected out of the cell because of this switch in the sodium-potassium pump. Now that it's open to the outside portion of the cell, and now that the sodium floats away from the cell, it is now able to grab onto 2 potassium ions. The 2 potassium ions now bind to the sodium-potassium pump here. But in order to get ready to be transported into the cell. In order for that to happen, though, that phosphate that was initially attached to the sodium-potassium pump needs to be removed. When the phosphate is removed, the sodium-potassium pump switches again, pumping the potassiums into the cell and leaving it open on the intracellular side again to start everything over. If you want to hear that explanation in a little bit more detail, you'll be able to see it on the video when it's done being uploaded. By the way, the video is about halfway done being uploaded. I'm uploading it in parts. It should be done hopefully by tonight, and I will send everyone an email link to it sometime really late tonight. Okay. Moving along. The only other pump that you have to worry about for this exam is the hydrogen-potassium pump. We're not going to have time to go over it now, so I do suggest that you guys review the hydrogen-potassium pump. I give a couple questions to help you study the 2 types of pumps here listed here. You can review these questions that are on Canvas. Whenever you see this pump here, though, just ignore it. That's old information. It's called the hydrogen ATPase. So anything referring to that, just ignore it. This is a little bit of old information. Yes?
Do you have those answers to these?
I would like to, and I will try to, but I'm very behind on a lot of stuff. At minimum, you guys have the videos to watch, which will cover the most essential things. I will try to post the answers to these things if I can. Yes?
On one of the practice exams, it had a question about